Hello, good morning, everybody, and thank you for attending this talk. As I've already been introduced, I'm going to be talking about MongoDB and Gene databases. And first off, before I start talking and go on with the talk, I'd like to thank the organizers. I'm really excited to be at the first PyCon. I would like to thank the organizers for having me here. I'd like to thank Google for sponsoring the diversity program, which has enabled me to come here and speak to each and every one of you. So thank you. Moving on. So I would like to just introduce a little bit of what I've done. I've been a free software volunteer from 2005, which is when I started my journey with Debian. I wrote a Sanskrit locale for Debian blox. It's in the blox package. I founded Ubuntu Women. I volunteer with a number of women's organizations like Linux Chicks, Sisters, KDE Women. I've been a list administrator, and I've enjoyed understanding how diversity actually works, why it is so hard for women to be in the community, in the free software community. When I started out, there were very few women, but now it's changed a lot. And I've done, as part of Google, I worked with the sisters. I did some Mailman code. We worked on 2.1.10 version that is pretty outdated, since Mailman is on 3x now. And I did the patches, testing, and release project for Sisters Mailman. And I also volunteered with the Scripps Research Institute in California, which is for whom I've done the MongoDB project. So you can, if you're interested, you could just check out all the work that I've done. I've listed it there. The Scripps Research Institute. So basically, the Scripps Research Institute is a research organization in California. They work a lot on gene databases, bioinformatics stuff. They do a lot of research in, they run the, so I worked with the Su Lab, which is a part of the Scripps Research Institute under Dr. Andrew Su. And they do a lot of, how do you say, gene wiki profiling. They have gene annotation databases in which researchers, biologists, and practically anybody who's interested in bioinformatics can go update the wiki. You can get data. If you're a student, you can probably just read about it. They have a few 30,000 pages on the Gene Wiki. They also run a gene annotation database site called the BioGPS and MyGeneInfo. So those two, the last two, were the ones that I've worked on. And if you check out both these sites, you'll find out that BioGPS is basically a site that allows you to do gene annotation. It allows researchers and biologists to go and get data on gene annotation, protein function, gene ontology. You could do protein, you could get wildcard queries, and you could get that information. So a lot of biologists and researchers, they use BioGPS in their everyday work. And MyGeneInfo is the site that powers BioGPS. So that is the one that I worked on. So as I've already gone through this, so it's basically, BioGPS is a customizable REST interface. It allows you to get gene ontology, gene annotation, gene symbols, protein functions, make wildcard queries, get asymmetric IDs. You could get mRNA sequences. You could get protein sequence alignment sequences. Basically, just anything that a biologist or a scientist requires. And moving on. So the server stack that we used is Ubuntu 12.04. Mercurial, because it's Python, and we run the full site on AWS. So currently, the site is running on CouchDB, Tornado, Python, and libraries that we have are BioPython, NumPy, and PyES for Elasticsearch. So I'm porting it from CouchDB to MongoDB. So that is what my project was all about. And the MongoDB stack that we are using is running on we use PyMongo, which is the driver, and MongoKit, the object data mapper. So moving on, this is basically the project, what the project is all about. So as you can see, the whole idea is that we collect external gene annotation data from different sources. So when you say sources, we have multiple databases from which we pull data. So it could be Ensemble, it could be Anthres, it could be data about homologene, it could be about a rat genome. So what happens is that, for example, a rat genome site would have data on gene ontology, it could have data on gene sequencing or gene annotation, but it won't have any data about phenotypes. That I need to get from a different database. So the idea was that we will collect all the data from multiple databases, put them in a single MongoDB document, because Mongo allows us to store data which is not exactly, which does not have sequential form of storing which you have in an RDBMS. We can store data which have different fields, which have different formats. So, for example, in certain databases, we have data which comes in a text format. In some places, it comes as a CSV file. In some places, they have stored it as a MySQL document. And we pull that data, we convert everything into a JSON file, and store it in Mongo. So the idea is that we collect all the data, and we have source scripts which we use to collect the annotation data, and we convert them into JSON documents. So for example, if I have 1017, that is the ID number in Mongo, I could probably check whether you have, what symbol you have, what is the name of the particular protein, and what's the symbol, corresponding symbol for 1017. So probably in a rat genome, it could be CDK2, but in an Anthrax database, it could be something entirely different. So the idea is that we bring all the data that corresponds to the gene ID number 1017, merge them in one single document, and then put them on BioGPS so that the researchers could probably go and check what is the latest. Because every time Anthrax updates their database, rat genome might not do it, or Reactome might not do it. So the researcher has to go to 3 or 4 different databases, do the research, come back and merge that data and see whatever it is that they're searching for. I'm new to biology. I've learned biology only through this volunteering, through this project. So I'm not exactly sure what the biologist does or what they're searching for, but I do know that when you have multiple databases with different types of data for the same gene, they have a problem, and that's the problem that you're trying to solve. So we are trying to put each of these IDs and merge these documents, collect them, and store them in MongoDB. So we have in the collection part, once we've done that, we have daemons that will run at a periodic interval. This entirely depends on the researcher's research, what they're trying to do. So sometimes they do it on a daily basis. If Anthrax updates their database every day, then the researcher runs the cron job that I've written, and he gets or she gets the data on a daily basis, and then it gets dumped into the MongoDB, and they're able to continue with their work. If they run it, sometimes the source scripts are run on a weekly basis. Sometimes it's on a monthly basis. There are some databases which don't update for a few months altogether. So that's what we look at. So the structure, as you can see on the right-hand side of the screen, is that we have a source script that runs on a periodic base, and we can add source scripts later so that they can have annotation data, they can have mRNA data, they can have protein sequences or phenotypes data. So depending on what the researcher requires. So this is actually a work in progress. So I'll be working on this. Since right now I'm at Hacker School, so I'll be going back to work on this in January again. So, right now we have for gene annotation and gene ontology. For the other things, I'll be probably writing scripts much later. Then the second part is the architecture. So earlier, as I mentioned, that's the URI for the Sioux Lab. You could probably just go there and check out all the projects that they have. It's pretty interesting. They have migrated a lot of their proprietary software into free software. They were part of the Google Summer of Code this year. And they did a bunch of games and Gene Wiki. So these were the projects, about 8 or 6 projects were selected in Google Summer of Code. And so the old architecture, as I mentioned earlier, it was in CouchDB. So, and everything, the data loading, the indexing, the searching, all of this was done as a single file. So we broke down the architecture. I've separated them into daemons, data building, data indexing, and data loading. So the goal is that we collect this gene annotation data from different sources, and then we merge them into the gene source collection in MongoDB. And then we use the Amazon Elasticsearch, which allows us, so that's the indexing part, where we allow the researcher to search based on wildcards, based on gene ontology, or whatever it is that they require. Then the backend MongoDB framework allows me to index all these documents in a periodical format as and when the biological sources are updated. Then, so if you can see, the daemons are for running all the scripts that the researcher requires. It could be on a daily basis, it could be on a weekly basis. So they will decide if they want a particular Anthrax database, and they want it on a weekly, say, next week or 15th of November. So they can just go there and run that particular daemon. It's just a single command that they have to do it from the command line. They can do that. And it will automatically take that, build the source, do the indexing, and load it for them. And it will, on the BioGPS site, this will be served for the researcher. So if you check out the code over here, the data loading, this actually allows the register sources module will allow the plugin architecture. It allows a metadata like collection name. It allows me to get the name of the file that is being processed, the document structure, and it will output all of this in each of the collections that are there. So we have around 9 collections in so far in Mongo. You'll see that as we go across. So now we come down to the building part. So again, when I'm building this particular database, I'm combining the individual annotation data in one single document. So for each of them, they are in different, they may be having different fields, they have different data. The only thing common between each of this is the gene ID number, which is 1017, or it could be anything. Now what happens is this becomes a big challenge because certain databases have different field structure. As you can see over there, some of them have just the name and the symbol. Some of them have unigene for the same ID number, that is the gene ID number. And some of them have a name, symbol, and a unigene. A regular RDBMS does not let us combine these kind of documents, whereas in MongoDB, which is why that's the advantage of having a NoSQL database. So I'm able to get databases, collections which have different structures, which have different fields, put the data together in one single file, and I'm able to serve this full thing so that the researcher can, get all the data that they require. This is not possible in a regular RDBMS, and that's one of the biggest advantage. So each of these combined databases sometimes run into 30, 40 GB. It depends. Sometimes it's even bigger than that. So just getting them to, how do you put it, running it on a local machine was really hard for me. I had a tough time, and I had to use their, the Scripps Research Institute servers to get this work done, because my machine does not have that kind of resource and capability to process 40 gigs of data just for a single database. And I had around 9 collections where I was updating data, so that really made it very, very difficult for me. Then, so if we move on now, if you see this figure, this actually is the architecture. I'm not sure if you're able to see this. Okay. Okay, so here you can see that a GeneCron Pi, yeah, if you can see, this is the architecture of the whole project, how it works, how we pull data from NCBI, the Anthres, Ensemble database, and we are able to use the cron job, which keeps a check on the scheduled time. It checked the file dl-anthres.py executes and downloads all the gzip files into a data dump folder, which I showed you earlier. It dumps them according to the date on which the cron job was run. It brings all the data that is available there. It needs to have a timestamp. So that is one thing that it does. It allows the researcher to go back and forth between two of the databases that they have pulled. Then once all the files are downloaded, it imports. There's a parse file for GBFF files, for refsec, it could be for annotation. So we have scripts that parse all this data. It again puts them in the data dump folder. Then the dl-anthres.py, that file, then controls the program for execution to the parser, which is the refsec GBFF file. And all the 5 refsec files process and create new text files as per the record in the gene bank. So it uses, as I mentioned earlier, we use BioPython. So there is a library called BioSecIQ, which reads the data bank. It formats, parses, locates each gene ID, gene annotation, gene ontology, and it lists them according to the gene ID in the record in our collection. Then after that, once we've created the GeneDoc source database, I run the data load script, and that will allow me to index anything that the researcher asks. So the PyES library, that is an Amazon library that is provided for searching. So we use a lot of their files. It allows me to combine and serve this data and support all the rich queries that is required. Then, okay, so if you see the data indexing part, this is, just a moment. Okay, so if you see some of the code, you will find that it's highly nested dictionaries and structures that we have. So the file, with the data indexing files will flatten the dictionary because they are so deeply nested that sometimes pulling out the data is very difficult. So this particular, it's a huge file, but I've just taken this part to show you that we can flatten the structure, and then we are able to then merge, we can flatten the structure, and then we are able to do the search for the indexing part of it. So we get the metadata, we can get the mapping, we can get it on the basis of the keys on the ontology, depending on the structure. If it's a protein kinase, then we can define what are the fields that they have used for that particular collection in Anthres or in Reactome or in any other database that we have. Then after that, this is a list of the collections that we have. So as you can see, there might be some data, like if I go back over here, there might be some data which is there in the Anthres Go database, but it's not available in a homologene. So each of this particular, for when I'm searching and I'm indexing the data, I need to flatten the structure because the structure that each of these databases uses is very different. A homologene database has a completely different file structure. When I drop that in my collection, it does not necessarily match what is there in a Unigene database. It does not match what is there in a gene info database. Oh. I'm sorry. There's quite a bit to go on, but OK. So I'll just quickly end this talk by saying that you could just go and pull the code from the URI. Did everybody get the URI? It's bitbucket.org sulab. So you just go and check out the code that we have. GeneDoc Hub is the code repo. So my code would be in bitbucket.org swaksha/GeneDocHub, so that's my branch. It still needs to be pulled into the core repo. So that's. I'm so sorry that there's quite a bit that I could discuss and talk about, but we've run out of time. So I'll just say thank you very much for listening. And if anybody has questions, you could please talk to me after after this is over. So I'm around. So thank you very much. I'm really sorry about it.